All right, so welcome back to the Classics in Immunology Journal Club. And before we get started, don't forget to like this particular video and subscribe to the channel if you want to, and also click on the bell so you get notified. I don't know why you have to do all these things, but anyway, click on the bell. Go and scroll down on the YouTube site, and you can find all my social media stuff and my website. And if you go on the website, click on the writing tab, and you'll find all the classic papers there. It took me a while to collect all those things and put them on the website, and I'm still finding that I need to revamp everything now and then. So a lot of work went into that whole thing. So check it out. So today we are a little over 30 years ago in 1991, and we're going to review a paper that is one of my favorite papers because it's one of my papers. And the authors are Stefan Ehlers and Kendall Smith. And the title is The Differentiation of T-cell lymphokine gene expression, the in vitro acquisition of T-cell memory. And it was published in the Journal of Experimental Medicine in January of 1991. Now, the background on this paper is that Stefan Ehlers was a postdoc who came to my lab from Berlin, and where he'd been in a laboratory that was an infectious disease lab, and they were experts in studying tuberculosis. And tuberculosis is a particularly interesting bug to study, in that the immune inflammatory response to the TB organism is complicated and so forth. And so Stefan got all excited about this over in Berlin, and he loved Berlin. It was a great big city, and it was before he trained there mostly before the wall came down, and so it was quite a place to be. And so, but he needed to get another postdoc, and so he was looking at my laboratory, and he was also looking at the laboratory of Steve Rosenberg down at the NCI, who was excuse me, trying to cure cancer by immunology. And so Stefan came to visit me up in Hanover, New Hampshire. And so he came from this big city, cosmopolitan and so forth, to this tiny little town, 5,000 people, residents, and 5,000 university students. We discussed all kinds of things, and I took him around and showed him around the town and so forth. And after he was ready to leave, he said, the only thing that bothers me about Hanover, New Hampshire is that, so what do you guys do here if you're not working? And I said, well, that's simple. We just work all the time. And he told me, and I told him that I said, don't go to Steve Rosenberg's lab if you're interested in really becoming someone. I said, you'll just be a cog in his machine. Come here, and it's much better environment, and it'll be fun. And so he did. And so that was good. He had become interested in the phenomenon of immunological memory, which of course is the basis for the immune system, for immunity, which means, it's a Latin word that means that you're exempt from disease. And it's also the reason that vaccination works and can protect you from infectious diseases. And since he came from an ID kind of a place, that was foremost in his mind. And I said to him at the time, when he interviewed, I said, we haven't been really into the mechanisms of memory, although we study what we've been studying is cellular proliferation, which is one of the hallmarks of the whole thing. And so if you come here, we can discuss it, and we'll see what can happen. So he did. So in the introduction of this paper, so he worked on this project, on this memory project, for 2 years. And this was his single paper out of my lab, which after reading it over again, 25, 30 years later, I said to myself, well, this is a really good paper. And it's really Stefan's contribution to the whole scene. In the introduction of this paper, starts with a definition of immunological memory. And it states that memory is the hallmark of the immune system, and it's responsible for immunity. That's why you become exempt, because you have immunologic memory to prior exposures of bugs. And it's also responsible for the efficacy of vaccination, because vaccination is just an artificial way to generate immunologic memory. And the further definition of memory is that it's the capacity of the host to respond more rapidly and with much greater intensity to secondary invasion by a microbe versus the first time around. So rapid and more extensive. And that's very important. The question is, how does the immune system generate that kind of a response? And the whole thing is based on Sir MacFarlane Burnett's classic paper in 1957 in the Australian Journal of Medical Research on his clonal selection theory of immunity. And it's also, the whole idea is, and it had been worked out for at least 20 years, maybe 30 before this time in 1990, in the B cell compartment, it had been clearly defined at that point in time that in addition to the fact that the B cells underwent clonal expansion during the primary immune response, they also then differentiated. The cells differentiated. So there's two aspects of hematopoiesis and immunopoiesis. There's cellular maturation that takes place before the cells leave their point of origin, usually in the bone marrow, but also in the thymus if you happen to be a T cell. You're immature until you undergo a maturation process that allows you to be recognized as a bona fide participant in the immune system. But you're still not differentiated yet. Differentiation is a second step whereby the cells acquire new functions. And immunologic memory is wrapped up in the capacity of the cells to acquire a new function in the B cells. So the B cells clonally expand after they get an antigen stimulation. And we've been going over in the past few sessions of our classics in Journal Club in how the B cells do this. It was established by 1990 that what the B cells do is they undergo a differentiation process that allows them to secrete antibodies, secrete immunoglobulin. So they go from a non-secreting cell to a secreting cell, secretory cell. And they also change their genes around such that they change the immunoglobulin molecule that they're secreting from the initial one to a secondary one. And we call that class switch recombination, because it's a whole genetic mechanism that we haven't really gone into quite so much in the Journal Club yet. And they change their heavy chain from a IgM heavy chain to an IgG or an A or an E. And those different chains of the heavy chain dictate how well that antibody is going to be, or how that antibody performs in the immune system. In addition, they have what they call somatic hypermutation of the antigen binding region genes, so that it's selected out for a very high affinity interaction between the surface immunoglobulin molecule and the antigen, and that selects out for clones of cells that have the highest affinity for the antigen, and that selects out for responding to very low concentrations of antigen. All of those things help establish the B cell memory response in the whole thing. Now, T cells, they didn't know T cells don't make antibodies. So they don't undergo class switch recombination of the T cell receptor, and they don't undergo affinity maturation of the T cell receptor either. Because the T cell receptor is not the effector molecule of the T cell system. Rather, the effector molecules were the ones that we've just been describing in the past 10 years, beginning around 1980, and those are the lymphokines or the cytokines or the interleukins. And these molecules, as we've seen, help the B cells. Because I always like to say that B cells are a little stupid and they don't know what to do until they get help from the T cells. And so the lymphokines have all kinds of functions in the T cell immune response, in the cell-mediated immune response. And in addition to the soluble molecules that the T cells make, they also make molecules that participate in the killing mechanism so that the T cells can directly kill target cells, which incidentally, and now that we're in the COVID years, is what allows the immune system, if you become infected. Question is, how do you get rid of those bad bugs? And the answer is the T cells do it, and they do it by creating an inflammatory response, by making cytokines that are inflammatory molecules, and they do it by expressing killer molecules that actually kill the target cells and rid the host of the bad bugs. So Stefan and I, as we discussed things, said that the difficulty in trying to study immunologic memory is the fact that the T cells all look alike. And you can't discriminate between a naive or a virgin T cell, anagenically virgin, versus an experienced T cell. A lot of people up until this time, in the past maybe decade before this paper, had been trying to take cells out of primarily, experimental animals, mice as well as rats and so forth. But also people studied human lymphocytes as well. And trying to say, well, maybe there's a surface marker that's specific for a naive cell and a surface marker that was different and specific for a memory cell. And the problem is and then you could use those surface molecules to separate the cells, and then study the naive cells and study the memory cells. Problem is that depending if you're using very sensitive biochemical and molecular techniques to try to understand the differences in these two different cell populations, if you have any contamination of one versus the other, you'll get, it's garbage in, garbage out. You won't be able to really tell the difference between the naive cell and the memory cell. And Stefan tried to do this for maybe 3 to 6 months after he first got there. Couldn't convince himself that he could really tell the difference, and that this was a problem that was insurmountable in terms of the experimental techniques that were available at this particular time. So he decided that the way to really attack this problem was to try to take the only really biological definition of a naive cell was a cell from a newborn animal or human, because a newborn has not been exposed to antigens in utero for the most part, and it certainly hasn't been outside in the environment, so it hasn't had any environmental exposure. And so it turns out his wife was a social worker in one of the local hospitals. And Stefan decided, instead of going to our university hospital and trying to get cord blood, umbilical cord blood lymphocytes from recent baby deliveries, he went over and sweet talked the nurses in the community hospital, and they joined into his project, and they'd call him at 3 a.m. and say, we just had a delivery, and he'd hop out of bed and get in the car and run down to the hospital and collect the cord blood samples and then take it back to the lab and start working on it. And so he did that for the rest of the time, basically, that he was in my lab. We never saw him because he was in the delivery room. I'm just kidding. So then we needed a readout for the cytokines. We wanted to study the cytokines. We didn't really study the killer molecules quite so much at that stage yet. And ultimately, it turns out, and the other thing was that we were limited in terms of the numbers of cells you could get from each baby delivery, the cord, and from the placenta, really. From the umbilical cord. And so just at that point in time, the polymerase chain reaction had been developed and was available to study. And we, because of the fact that the PCR, which is used to amplify the COVID virus so that you know whether or not you've been infected or not. The great thing about this is you can undergo 20, 25, 30, 35 cycles of amplification, and therefore you can detect it's a very sensitive technique to detect very small concentrations of molecules. And so he developed the PCR technique in our lab and started going from there. So that the experimental question that he attacked was that are there any differences in the expression of the genes that encode the known cytokines, lymphokines that had been identified at this point in time between the naive cells and the memory cells that he could get from basically normal adult blood, because adults being around for 20 plus years, they had memory, immunologic memory, and also the cells that are responsible for that immunologic memory. And so he set about working on these sorts of things. And so in the materials and methods of this paper, by this time we had multiple monoclonal antibodies that had been well characterized in other labs and that were helpful in these kinds of experiments. We had an anti-CD3 molecule, which was a very special molecule reactive to the CD3. And as you recall, when we went through the T-cell receptor characterization, the CD3 components are actually several chains that are the signaling chains that transfer the signal from the outside of the cell to the inside of the cell and activate the signal transduction pathways that ultimately result in new gene expression in a T cell and IL-2 production and IL-2 receptor expression and so forth. There's also a pan T cell marker called CD2 that Alice Reinhertz and his group at Harvard had already described and detailed, and so we had available for that antibody. And then we could buy at the store OKT3, 4, and 8, anti-IL-2 alpha chain receptor alpha chain, and that we also got that from Ehlers. So friends are important in this business to get other reagents that you might not have in your own refrigerator. We got recombinant interleukin-2 from Takeda Chemical because they produced a large amount of that, and we got that through Koichi Kato, who had been the person I think that we talked about before that helped us define the IL-2 receptor, second chain. Now, Stefan worked out an elaborate cell purification protocol because he wanted to obtain pure T cells that he could activate via the CD3 molecules on the cell surface that did not require any accessory cells. And we didn't want to get involved in the CD28 pathway. We wanted to be able to activate these T cells only through the TCR and CD3 molecules, and using the special antibody that Ellen Vitetta had originated down at the UT Dallas, Texas Southwestern. And that was called 64.1. It was just the number of the monoclonal antibody. And so to get the cells purified, so we got away from all the accessory cells, and we could say, okay, we've got really pure T cells here, and we really wanted to have only CD4 positive T cells to focus on in that business. We first take peripheral blood mononuclear cells. We would take the blood and separate the PBMCs over a Fecal-Hypec density gradient, where a band of white blood cells that are all mononuclear cells, and getting rid of the polymorphonuclear leukocytes that pass through the Fecal-Hypec. So you get a band of mononuclear cells that you can pick off with a Pasteur pipette. And then we took those cells, washed them a couple of times, and put them over another density gradient made of 65% Percol. And again, the lymphocytes would band out on that. And then to get rid of the accessory cells, the so-called the macrophages primarily, or the residual PMNs, the phagocytic cells, Stefan got a method from the literature where we incubated the cells with leucine methyl ester, which is taken up by this phagocytic cells. It would then allow you it would kill off the phagocytic cells, and you could then take the residual cells left over and spin them out and wash them and so forth. And then we also got rid of all of the phagocytic cells would have FC receptors on their cell surface, and so we would incubate the cells on petri dishes that had been coated with immunoglobulin, with antibody, and those immunoglobulin molecules would latch onto the FC receptor on the monocytes and the macrophages, and they would stick to the plastic, and then you could wash off the non-adherent cells and take them, which is what we did. And then there was another thing that Stefan introduced, which was a product that we could buy at the store, which was called Lymphokine, and that had antibodies to HLA-DR positive cells, which would be the B cells and the monocytes. So that would take out both the monocytes as well as the B cells. And it would also, it had antibodies to the polymononuclear leukocytes, to the natural killer cells, and to the CD8 positive cells. And that was important because these cells could also make cytokines of various varieties. And we didn't want to have the results contaminated with these cytokines coming from gene expression by these contaminants, is what we call them. So then, in order to establish cell purity on these cells, we went through several different tests to see how pure they were. We found that in the cord blood cells, we could get close to 99% CD2 positive cells. That was the pan T cell marker. 99% CD3 positive cells, 95 to 98% CD4 positive cells, 0 to 1% CD8 positive cells. And we got exactly the same results in the cord blood cells versus the adult T cells from just a normal person. In order to stimulate the cells in culture, before we assayed for the messenger RNA of the different cytokine molecules, we would take microtiter plates and use the tiny little wells, 400 microliter wells in the flat bottom microtiter plates. We'd coat the bottom of these with a goat anti-mouse immunoglobulin. So it's an antibody to a mouse antibody. And we would put the cells down onto there, and we would put in a soluble form, the 64.1 anti-CD3 antibody. So the anti-CD3 antibody would be caught on the bottom by the mouse anti-immunoglobulin molecules, and then we would put in the cells. And the reason that is important is, and I had to ask recently my friend Ehlers Reinhardt's, who's been studying the T cell receptor for the last 50 years, and he's determined that this is a mechanoreceptor. In order to trigger the T cell receptor, what you have to do is put a force onto it, torque. And by having the first antibody onto the plastic, the second antibody on top of that, and the cell then gloms onto that, it's really a lattice work. And as the cell tries to move around a little bit, because it has muscles, it has actin and myosin, every time it does that, the T cell receptor gets tugged on, torsion. And that's the generation of the biochemical signal for the T cell receptor to become activated. We didn't know that at the time, but now we know that. I thought I'd just sort of throw that in, but since we do know it. Then in order to study the proliferation of the cells, we do the time-honored method of culturing the cells in the incubator at body temperature for several days. And then at different time intervals, we would take out the cells or we would pulse the cells with tritiated thymidine taken up into newly synthesized DNA. And then we could that's how we could monitor and quantify the proliferation. we could also take supernatants from these cells at different time intervals, and we had various assays for the lymphokines that we were interested in. And we had the IL-2 bioassay that we'd originated in my laboratory. And then by this time, there were biotech companies selling kits that determined by the enzyme-linked immunoassay method for interferon gamma and GM-CSF and so forth. And so that's the other way we could measure these cytokines. But the final thing was the PCR mechanism that I mentioned for the messenger RNA. This was quite a long process, but after the soups were harvested from these cultures, we could wash the cells in the microtiter plates and then add chemicals to extract the RNA. Then we could use the PCR machine that would cycle at different temperatures through 25 cycles of amplification. And we would amplify up the messenger RNA molecules in the cells. And so it was a very, very sensitive technique. And we could then take the RNA or the cDNA at this stage of the game and run them out on gels. And then we could then add cDNAs for all the different lymphokines. And we could then stain those things with lithium bromide. And so that's what I'm going to show you as we go into this now for the message of these different cytokines. And so let's do some screen sharing here. And there's 11 different figures in this paper, but they're pretty easy to deal with. So the first question that we were interested in is whether or not we could activate these cells by this elaborate business with anti-CD3 64.1 antibody, and whether or not we could get proliferation of these cells in both the neonatal and the adult, and whether it would be equal. And the answer to that question was yes. This is thymidine incorporation here. This is increasing concentrations of the antibody to CD3. And as you see, the concentration goes up. These are the cord blood lymphocytes, T cells in the open squares, and in the solid squares are the adult T cells, and they're absolutely essentially identical. So that part of the system worked. So then the question was, so we wanted to look at the proliferation of these cells and compare them, the purified T cells, to the whole mix of mononuclear cells, and to see whether or not they were equal or not. And so in lanes 1 and 2 here, we're still looking at thymidine. We're still looking at proliferation. Lane in 1 and 2 is these are adult T cells, purified, nothing else except T cells, cord blood T cells that have only been given soluble anti-CD3 and that you get no proliferation. And here in lanes 3 and 4, we have adult and cord blood T cells that have been cross-linked with the Petri dishes and the mouse antibody. And then in 5 and 6, we have in lane 5, we have neonatal T cells activated through PHA and PMA, which are known T cell stimulants. And then we have mononuclear cells, neonatal mononuclear cells. These are purified T cells, and these are mononuclear cells activated with PHA. And you can see that they're all basically error bars. They're all equivalent in these situations. So that was good. In figure 4, we're looking at the production of interferon gamma and GM-CSF. And what we see here and so this is in picomolar. These are done by ELISAs on the supernatants now. So we have adult T cells, neonatal T cells at 24 hours and at 48 hours, and there's no gamma interferon. And then these are adult T cells at 24 hours and 48 hours, and there's easily detectable gamma interferon. Now, GM-CSF was a little different in that at 24 hours, it was negative, but there's detectable GM-CSF by 48 hours from the neonatal cells However, by comparison, there was more GM-CSF in the adult cells after 24 and 48 hours. Now, the thing which isn't shown here is other experiments that Stefan had done had shown that the GM-CSF coming out of these cells at later time points is because it's an IL-2 induced gene. It's not solely dependent on the T cell receptor. And I think that'll come out in some of the other data. So then, once those experiments were done, figure 5 is the PCR technique applied to adult cells on the top and neonatal cells on the bottom, and then looking for IL-1, 2, 3, 4, 5, 6, interferon gamma, and GM-CSF, and the IL-2 receptor alpha chain on unstimulated cells before the experiments were started, before the stimulation, and basically they're all negative. This is beta-actin as a control that there wasn't something wrong with the DNA. So here in figure 6, this is after 3 hours of activation of adult cells versus the neonatal cells. And here it's clearly, it's after 3 hours after activation with the 64.1 anti-CD3 and the cross-linked on the Petri dishes, we can see IL-2, we can see IL-3, IL-4, IL-5, don't see IL-6, interesting, interferon gamma, GM-CSF, and then the alpha chain of the IL-2 receptor. So those are the adult cells. In the neonatal cells, look at that, we only see IL-2 and the IL-2 receptor, alpha chain. So that was only at 3 hours. And so the question is, if you do multiple time points, will you see more? So figures 7 and 8 are very critical in this regard. We're seeing 4 different preparations here. This is after 6 hours, 9 hours, 24 hours, and now 48 hours. And this is PCR amplification of the message of adult T cells at various times after stimulation of anti-CD3. So we're looking at 6, 9, 24, and 48 hours. And you can see that basically the patterns are very, very similar. Interleukin 5 that was negative at 3 hours is clearly positive by 6 hours and 9 hours, and even more so. So it just took a little bit longer. IL-6 was still negative, and the interferon gamma was positive throughout, and as was GM-CSF and the alpha chain. Now here's amplification of the neonatal cells at these same time intervals, 6, 9, 24, and 48. You can see that there's IL-2 is positive, and GM-CSF comes up at 24 and 48 hours because it's an IL-2 induced gene rather than simply a T cell receptor induced gene. Figure 9 then is looking at neonatal T cells that have been activated only by PHA and PMA as alternative activators that cause the cells to proliferate, but they don't make any of these other cytokines. And then the real question was, after all of these experiments had been done, can you use this, the inability of the T cells, neonatal T cells, to express these other lymphokine genes after they've been activated? What happens if you allow them to proliferate for a week or two, and then we knew that what would happen is the cells would stop proliferating after about 10 to 14 days. And then you could, with adult cells, you could then re-stimulate them through their T cell receptor, and then they would begin to grow again. And so that was the system that Doreen Cantrell had worked out that became so important for our whole laboratory. And so we did this exactly the same kind of an experiment. And in figure 11, then we have neonatal T cells after first a primary immune response where they were activated with anti-CD3 for 48 hours and then washed out, and then kept in IL-2 for 7 to 10 days, I think, and then re-stimulated through the T cell receptor with the anti-CD3 molecule and so forth. This is time 0 on the secondary stimulation. And then this is 3 hours, 9 hours, and 48 hours. And you can see that now these neonatal T cells that couldn't do anything before are now just going like gangbusters with all of the cytokines, just like an adult memory experienced, no longer a naive T cell. This is a no longer naive T cell, essentially. And if you don't re-stimulate them and you keep them on those same days, they don't express any of these genes. And so that's it for the data in this paper. Stefan was exhaustive in the way he did his experiments. He would do them over and over and over again to make sure that they were really true. I mean, he had some 20 exactly the same experiments, 30, 40 different experiments. He just didn't do them 3 or 4 times. He did them 30 or 40 times. So in the discussion of this paper, actually, I think I recommend reading this paper because this is it's an example of a very rigorous scientific approach to this particular question. And the discussion of this paper, we start out by saying, or Stefan essentially says, that to remind everybody that during a primary antigenic exposure, there is a change that the immune cells undergo that is responsible for the generation of immunity. It's crucial in that change that happens that the cells have the ability to remember the first antigenic exposure. And so this whole paper is simply the first step in defining the molecular events that develop after a single stimulation. Now, in order to do this, he had to establish an experimental system to be able to distinguish between naive and adult T cells, as I've said, and therefore using the biological definition of naivete, of virginity on the part of the cells, the best and only system available was to take cord blood lymphocytes. Therefore, what he was able to show was a clear qualitative or molecular difference between the primary and the secondary stimulation. And in this instance, CD4 positive T cells can express all of the cytokine genes that we could measure in this particular series of experiments. And therefore, the cells coming out the other end had the memory. And in medical terms, we call that an anamnesis, which is a Greek word that is translated as remembers. We use it all the time when we take a history of the patients. We call that an anamnesis. Then in the discussion of this paper, he goes through the several different points that needed to be rigorously established. The first point is that it was essential to begin with homogeneous, purified, naive cells, because any contaminating memory cells in the naive population would just obviate any of the results. You couldn't really interpret the results. And the cord blood was the only known source of all naive cells. And it was crucial to develop all those rigorous methods that he did in order to purify away contaminated accessory cells. And if you had any accessory cells there, what would happen if you used soluble anti-CD3s is that you would get activation. We just went through the whole CD28 molecular story in terms of the co-stimulation, and we didn't want to have anything like that involved in these experiments. It was also important to use at the other end a very sensitive method for the readout of the messenger RNA expression in these experiments. And so the PCR amplification of the message was really important. Now, he also qualified the whole interpretation of these experiments by saying that, okay, this is at the cell population level. We have thousands and millions, hundreds of thousands and millions of cells in these tiny little wells. And so we couldn't make any statements really at the single cell level then 30 years ago. Now we can do that. And so there's a lot of work to still be done on this whole story. The other point that he made was, there's a whole series of experiments that had been published in the 80s about different subsets of T helper cells that could be distinguished on the basis of their cytokine gene expression. And they called some Th1 cells and other ones Th2 cells. But that was only done in the mouse. When people tried to do that in the human, they couldn't make that distinction. And that was very important, and I think still has not been shown. Or in T cell clones that could be distinguished in that way. The explanation for the memory capacity to express all of these genes within just a few hours after activation of the memory cell, both in vivo, the adult cells, and also in vitro after a primary stimulation in vitro and culture. You can see why once you have that memory and you can express those cytokine genes immediately, it's a heck of a lot better than having to wait several days to be able to do it. And that's one of the reasons that memory is memory. One of the reasons that vaccines are effective, because of the fact that during the vaccine priming situation, you generate the memory. So the other thing that is interesting, I think, particularly now to read it now, 30 years later, is that we speculated, or Stefan speculated, that perhaps some of the pathogens that we have to deal with out there in the big bad world could impair the delay the development of the memory phenotype, the ability to re-express those cytokines so rapidly. And if so, then the total response would not occur right away. And that would allow the microbe to just sort of proliferate on and do whatever it wanted to do with no governorship over it. And of course, that's what the COVID virus does to the interferon gamma production and also the interferon alpha beta production by the cells early on after the primary infection. That inhibition of the interferon response allows the replication of the COVID-2, and it can take place for anywhere from 7 to 10 days to 2 weeks. And during that time, the person can be asymptomatic, which means that the symptoms only occur when the immune response finally kicks in after the organism is proliferated up to the point where you're crawling with these bugs. Then when the T cell immune response finally kicks in after 2 weeks of infection, then you have a war going on at that particular point in time. That's when the symptoms appear. And the symptoms are due to the immune response, and the symptoms are due to the cytokines, the pro-inflammatory cytokines, interleukin-1, TNF, and interferon gamma. I'm very pleased with this paper and these results. So subsequently, just to give you an idea, what's happened subsequently is, so Stefan went back after a couple of years in my lab, and he went back to his original lab in Berlin and stayed there for a couple of years or so and worked on things back in Berlin. But one of the things that happened to him while he was in Hanover was that he found a really nice condo apartment across the river, the Connecticut River, in Quechee, Vermont. So he was in the trees and so forth, and it was quiet, and it was just nirvana by comparison to the cacophony of a big city like Berlin. And he, after a while, he acclimated to that whole system or whole way of living. And so here he found himself back in Berlin, and although it was a cosmopolitan with all kinds of good restaurants and so forth, ultimately he was recruited to go to a smaller research institute up near Hamburg, in the north of Germany, called Borstel. And that was an infectious disease research institute that was studying TB. I mean, what could be better than that? That's what he wanted to find out. And so he had a whole career, a very productive career there, studying the mechanisms involved in the host response to tuberculosis and so forth. Ultimately, he became the director of the institute, and he's just retired a little while ago, in the last year or so. Meanwhile, back at the ranch in Hanover, we didn't follow up on this series of experiments because we were heavily involved in trying to understand new gene expression in proliferating cells, and cells that have been stimulated by IL-2 to undergo what we called cell cycle progression, replicate their DNA, and undergo mitosis, because that's where I had started out. So I figured this was Stefan's area here to continue and so forth. But I'm not aware of others who followed this paper, even now. And so if you know of other things that I don't know, please contact me. Send me an email, please, and because I'm very interested to find out. The thing is that the molecular mechanisms involved in this whole process, what happens after the initial stimulation of the T cell receptor, that ultimately allows a secondary stimulation through the T cell receptor to now activate all of these, the expression of all these other genes, is interesting, because it's basically in addition to proliferation, the cellular basis for the more rapid immune response in a secondary situation. So that's it. And thanks for listening. And don't forget to like the video and click on the bell and check out the website. And so we'll see you next week.